Hi guys, today's study tip: tell your entire household the dates for your exams. Why would you do this? Because you want support and you want everyone to know that you are going to be extra busy the week before your exam and definitely not bother you the day of the exam. But like I said, extra support. Maybe they help out uh, with various chores or cooking or just emotional support, right? Okay, so put it on your family calendar, put it on your significant other's calendar, put it on your roommates, housemates' calendar. Just let everyone know your final is May 20th. You are super busy studying. Okay, so with that said, you're almost finished. It's the time to make the final big push, put in that extra study time, and finish strong. You want an A on your final so you can get the highest grade possible on this class. Now, the final's not cumulative, as you know, and it's not worth any more than the other exams. So, um, don't put too much pressure on yourself, but put enough pressure that you study. Okay. Put an outline for this one because I think your textbook goes in a different order. And I know the Dr. Hayes goes in a different order. Okay. But this is what I thought flowed the best. So we'll talk about chromosomes. We'll talk about meiosis. We'll talk about spermatogenesis, fertilization, sex determination, and briefly hormones. Luckily for men, there's not a whole lot of hormone going on. There's no fluctuation. All right, so there's just a few hormones, just a simple regulation. When we get to females, it'll be a lot more intense with the hormonal regulation. Okay. All right, let's do it. I am drinking tea. So if you hear weird pauses or weird noises, that's what's going on. I'm trying to wake up. All right, so chromosomes. Somatic cells, as you may recall, are the cells of your body. They will all have two sets of chromosomes, so 46. And we call that 2N. So the way to refer to ploidy, or how many copies organism has. So our body cells are 2N, or another way to say that is that they are diploid. Okay, and the reason you have two sets is that one is from your mother and one is from your father. Each chromosome has this duplicate copy called a sister chromatid. So this picture down here, this is one whole chromosome. You don't normally see it like this. It would just look like one, right? So one from mom and one from dad. And you cannot tell by, in this picture here, this image is called a karyotype. And this is when you take the DNA from, this is human DNA, but you can do this with other organisms as well. And you stain it and you image it. But because these are molecules, they're, DNA is a huge molecule, so we're able to do this, but it's still a molecule, so it's still tiny. So you actually can't tell in this image that each of these strands is actually this, okay? And I know the way that these are paired up makes it look like, oh yeah, that's one and that's two, like this is one and this is two, but no. Each individual strand here in this image has a sister chromatid that you cannot see, okay? Because it's bound, it's tight, and again, these are molecules, they're small, it's hard to get that good of a resolution. So you just have to take my word for it, I guess. Okay, so one more time in this karyotype, this is showing one chromosome from mom and one chromosome from dad, or vice versa. We don't know which one's which. And each of those chromosomes has a sister chromatid, which is an identical copy. And I'm emphasizing that because the whole focus of meiosis to make gametes is to reduce the amount of chromosomes in each cell. So reproductive cells are called gametes. They only have one set of chromosomes, so it's either going to be mom or dad. And you're also going to lose your sister chromatid. And this is a haploid, and the other way to say haploid would just be in, so parentheses in, or one in, right? So in math, you don't have to write the one, but you certainly could if you feel better. Okay, so now let's review our meiosis. Okay, so recall that you start with one diploid cell, which is 2N. So this cell here is 2N. It has a copy of mom and dad chromosome, and each of those has a duplicate sister chromatid. So it's 46 chromosomes each with a sister chromatid. Okay, it is one chromosome. Don't add extra because of the sister chromatids. I think a lot of people get confused. So the sister chromatid is attached and is one chromosome. Okay, so you have a total of 46 chromosomes. They just happen to be, like, you could think of them as extra thick, right? They have the sister chromatid. Okay, then it's going to go through metaphase, right, where they go in the middle. And this is different than mitosis, where in mitosis they line up single file line. In meiosis, they actually pair up. And this is really important because the homologous chromosomes, the chromosomes that are the same code for the same genes from mom and dad, so that basically the identical chromosomes line up and pair up together, which you can barely see in this picture. It's very small, but I have another slide to demonstrate. And this is where a very important event occurs called crossing over. And this is one of the most important mechanisms that provides genetic diversity. So every time there's every single gamete is going to be unique. And this is why someone can have, I don't know, what's the most children anyone's ever had? I don't know, but let's just say you could have, try to be realistic. Let's say you have something like 10 or 12 children. They will each be unique individuals, not just because of personality, but because of genetics. You could take that a step further, and you could actually look at somebody's gametes and sequence them, and you would see that every single one has genetic diversity. Okay, so moving on, and we will talk on the next slide or two more about this. Okay, so metaphase 1 is where crossing over occurs. Then you go through anaphase, right? They get pulled apart. Chromosomes get pulled apart to opposite ends. And then telophase, you start making the nucleus again, and you get ready for the cell to split in two. And then you're left with 2 daughter cells. And because this is meiosis, those 2 cells have been reduced. So instead of having a copy from mom and a copy from dad, you have now split that apart. So now you have 2 cells, each one in. So you reduce the ploidy here, but each of those chromosomes still has its sister chromatid attached. And so we need to go through another round of cell division to remove the sister chromatids, or separate them, rather. There is another event here in metaphase and kind of anaphase that also gives you more variety is that you don't have to have, and you won't, it'd be the all of the chromosomes on one side that are going to go to this, and all the chromosomes on the other side that are going to go to this cell, they are not going to all be from mom on one side and dad on the other side. They can arrange in any arrangement. Okay, so you're going to get even more variety because of this, what we call, I'm forgetting the term. There's actually a few different terms that have been used, but basically there's no set arrangement of how the homologous chromosomes pair up. So some of them mom will be on one side and some of them mom will be on the other side, right? And so you will have a combination of chromosomes from mom and dad here, and that'll be different every time that you go through this process. Okay, so in addition to crossing over, you also have different arrangements or separation of chromosomes. Okay, now, but the main most important genetic diversity event is crossing over in metaphase 1. All right, so moving on, you have two cells here, daughter cell. So really this arrow should be pointing down here where it says daughter cell. Okay, but now you have two cells, each one N, so you reduce the ploidy from 2N to 1N, or you can just put N. And so now you only have one set of chromosomes in each cell. So either from mom or from dad, but not both. Okay, but they do still have their sister chromatids attached. Next, you go through all the steps again of metaphase, but because you only have one set of chromosomes, you're not going to have crossing over because there's nothing to cross over with, right? So now metaphase looks like what you're used to it looking like, where everything is in a single file line down the middle, and then they get separated, and now you can see the sister chromatids are being pulled apart. And so you're left with 4 cells, each of them genetically different from one another. So you have a total of 23 chromosomes, but they're by themselves. There's no sister chromatids. Okay, so let's simplify this. You start with one cell, which is 2N. It's a diploid. It has 46 chromosomes, each with sister chromatids. So I showed this, right? Sister chromatids, one from mom, one from dad. I'm not going to show all the steps because I'm not going to ask you about each of those steps, but I do want you to know that crossing over happens during metaphase 1. Okay, and then what do you end up with? You end up with 2 haploid cells, and what it got cut off, but it just said in. Okay, and then sister chromatids are still there, and so I showed that here. Okay, and then you go through another round of cell division, so meiosis 2, and that gives you 4 unique gametes. They're haploid, and there's no sister chromatids, right? They got pulled apart. Okay, so this is what you need to know. So what was that crossing over I was talking about? That is so important because it results in this recombination of genes so that each 4 unique chromatids result. Okay, so here's one chromosome with sister chromatids. Here's another chromosome with sister chromatid. Okay, so one is from mom and one is from dad. We don't know which one. Okay, and during metaphase 1, they line up, they pair up, right? And this pairing up of homologous chromosomes means that they can actually touch, and the top can and will, and the bottom can and will, but only for the ones that are right next to each other. The outside ones are too far away. They won't receive any of this action, okay? But the ones in the middle, they touch and they exchange genes. Now we show it here as one huge chunk, but it's actually going to be individual genes scattered along the whole length. And so you're going to have now you end up with these 2 chromosomes where each sister chromatid is different. You don't have any identical copies anymore. And then you're going to go through the rest of cell division, and by the time you get to the very bottom, each gamete has just one of these sister chromatids, and it's completely different and unique from all the others. So this is really cool. I suggest you could find a short little YouTube video that will give you a better visual of this. I did not think to do that at the time, but I have included lots of other videos for you. Okay, so Meiosis Summary. Here's a video actually from the publisher of your textbook, but it might have come from a different book, but it's the same publisher. Anyways, I like this series of videos. You've probably seen a couple already in this semester. So watch this on your own, please. Remember, you just highlight, copy, and paste into your browser. I know because you get the PDF, you don't have the hyperlink to just click on it necessarily, but you can still be watching all of these, and I expect that you are. Okay, so summary of meiosis, 2 rounds of cell division, so completely different than mitosis. You only have 1 replication event, and that's at the very beginning, and that's why you ended up with sister chromatids on each chromosome, right? So that was the replication. So you have 2 sets of chromosomes, and each of them have a sister chromatid because they went through replication. And then that doesn't happen again, right? Because we need to reduce our ploidy, and we need to get down to 4 gametes with just 1 chromatid. Okay, tetrads are formed during metaphase 1 only, and this is huge event that leads to genetic variability. It's a hugely important event, right? Okay, and then the result of meiosis, 4 cells with 23 chromosomes, each genetically different from the other, and only 1 chromatid. No sister chromatids attached anymore. Okay, so again, please watch this video. All right, so this is the male reproductive system lecture, and up until now we haven't talked about that. So, but I had to do that review because we're going to now talk about how our sperm made, and so you'll see the relevance very soon. Okay, so I trust you already know your anatomy, but I put this slide here in case you need to refamiliarize yourself with all of the anatomy. You should know all of this, right? When you go into nursing school, they're going to expect you know all of your anatomy that you learned. And when you take your nursing exam, you have to know all of your anatomy, okay? All right, so here is really the focus of what we'll be talking about today. So in the testes, you have your seminiferous tubules. So these kind of squiggly lines are the tubules. And within the tubule is where sperm production occurs. And so if you take one of those tubules and you look at it in a cross section, then you can see that what we'll be talking about. So on the outermost edge of the tubule are the germ cells, and then as it undergoes meiosis, which we call, for sperm, we call it spermatogenesis, it moves towards the center, which is called the lumen. So you will see cells in each stage of spermatogenesis, and then by the time you get to the center, you have sperm. Okay, so that's what we'll be talking about for the next few slides. So I put that here to remind you. Production, so spermatogenesis is the production of spermatozoa. So the sperm here in the lumen are not actually called sperm, they're called spermatozoa. And the actual production of spermatozoa is turned on when testosterone begins to be produced at higher levels, and that happens at puberty. Okay. And again, this is a cross section of the seminiferous tubule within the testes. Okay. So step 1 of spermatogenesis, begin as a spermatogonium, so that germ cell, spermatogonium, and it's actually enclosed or surrounded by a special cell type in the tubule. The seminiferous tubule has a special cell type called Serratelli cells. And these are what help to actually go through the process of spermatogenesis. So super important. So boys are born with lots of spermatogonia. So this very first germ cell, okay? And then at puberty, then they start going through all the stages of meiosis to make sperm, okay? So the cell has to go through actually mitosis and then meiosis. And so this is very different from women, okay? Women are born with all the ova, more ova than they'll ever use. Okay, but males have to continuously make more and more sperm. Okay, so they have to keep their germ cell. They need to keep one copy for themselves and make a new copy that will undergo a spermatogenesis and turn into sperm. Okay, so this outer layer is always maintained through mitosis. Okay, so the goal or the process is to make 2 identical cells, and we'll call one cell A and cell B. Cell A is going to stay, and it's going to continue to just go through mitosis over and over again. The other cell, we'll call cell B, will leave, right, and it will start the process of becoming a sperm. And so the cell that leaves or goes to, moves a little bit more inward, is then referred to as the primary spermatocyte. Okay, so the primary spermatocyte moves through tight junctions of the Serratelli cells. So in the tubule, moving a little bit more towards the lumen, it has to move through Serratelli cells. These cells also communicate and provide an environment for the spermatocyte that allows it to go through meiosis. Okay, so then it will go through meiosis 1 and is then referred to as a secondary spermatocyte. And of course, you start with one primary spermatocyte, you go through meiosis 1 and you end up with 2 secondary spermatocytes. Then you go through meiosis 2 and you end up with 4 early spermatids. And these spermatids don't have tails, right? They're missing all sorts of parts that they need to be considered sperm, right? So then they undergo spermiogenesis, which is the conversion of spermatids to spermatozoa. And during this process, they lose the majority of their cytoplasm, and that's when they become in that characteristic shape that you're used to seeing. So spermatozoa enter the lumen of the seminiferous tubule. They gain their tail, their acrosome, right? They've lost most of their cytoplasm, and then they will be moved into the epididymis. And they're not motile until they're in the female reproductive tract. Even though they have the tail and they have all the parts, you want to conserve all of that energy for the moment where you can actually get to the ovum. If the sperm were to start moving around before they were in the female reproductive tract, they would never get to the ovum, or they would run out of energy, because are just a single cell, and they all they have is the energy that has been stored within them, and once they burn through it, that's it, nothing more. Okay, so they actually will not be moving around, moving their flagella until they're in the female reproductive tract. So we'll be talking about that in a little bit later. Okay, so one more time. So here is the cross section. So imagine you take this circle, complete it. If you just look at one little spot, so here is the outside or the basement membrane, and then in here would be the lumen right in the center. Okay, so yeah, lumen of the seminiferous tubule here, wall of seminiferous tubule here. Okay, and you start out with that spermatogonia, remember, and then you go through mitosis. You keep one so that you can keep going through this process and making more sperm for the rest of your life, and then you let the other one move down and become a primary spermatocyte. And what I mean by become a primary spermatocyte, when it moves, it squeezes through Serratelli cells. It actually, now when it's in this location, it has a new environment, and it receives like chemical messages that turn on the process for meiosis to occur. Okay, and then it continues to move down towards the lumen. So now you go through the first division, and it's not shown, it doesn't show the second cell, it's just showing one. Okay, we're just focusing on one. But then you end up with your secondary spermatocyte, and that's where you've reduced from 46 to 23 chromosomes. But you still have your sister chromatids, so you need to undergo another division, right? And it doesn't show it here in these. This is writing from your textbook that I don't have the ability to change, which is why I was so careful to make slides for you that do show exactly what's going on, right? So if you go back and you can correlate this to spermatogenesis, then you'll see, okay, right. So at the end of the first division, even though you only have 23 chromosomes, they still have their sister chromatids. And then you have to go through that process again to separate the chromatids, okay? So this is nice because sperm, although they do start with mitosis, and that's not shown here, they do otherwise go through these steps, and you get 4 spermatids at the end, okay? Females are more complicated. That's just in every way true, okay? So let's go back to where we were. Okay, and then here at the end of the second division, you have your spermatid. So now you have 23 chromosomes, but just individual chromatids, no sister chromatids anymore. Now you need to go through spermiogenesis, lose the majority of the cytoplasm, and see you now have this characteristic shape of a sperm. You get your tail, you get some other parts, and then you are into the lumen and sent off. Okay, that's it. So one more time, spermatogenesis starts with mitosis, which is the identical copying. One of the cells will stay, okay? It will stay, and it will go through mitosis again and again and again. The other cell will move, the copy will move down towards the lumen, a little bit towards the lumen, right? And that becomes the primary spermatocyte. The primary spermatocyte goes through the first meiotic, so meiosis, first meiotic division, and gives you 2 secondary spermatocytes. Then you go through a second meiotic division to separate the sister chromatids, and you're left with spermatids, individual chromatids, spermatids. It's kind of nice how that language worked out. Okay, then you go through spermiogenesis to get the characteristic shape, right? Lose that cytoplasm, and get your tail and etc. Okay, I've put a couple links. This is a real short one from your textbook, and this is a little bit longer but walks you through these steps with really good visuals. I think it's from Khan Academy, this one. Okay, next. Oh yeah, okay, so mitosis, you have 46 chromosomes with sister chromatids. Then when you go through your first meiotic division, you will only have 23 chromosomes, but you still have the sister chromatids. And then when you go through your second meiotic division, you've separated the chromatids. And individual chromatids, spermatids, 23, no sister chromatids. Okay, so a mature sperm is called a spermatozoa. Well, an individual one's called a spermatozoon. Plural would be spermatozoa. Okay, and then what does that look like? Well, you have this special outer coat called the acrosome. We'll be talking more about that in a little bit. And then you have the nucleus. So the blue part contains the genetic material, right? Then you have this mid piece, which is really important in helping the function of the flagella, so that when the time comes, it will be highly motile. Okay, and then this is showing so this big, huge orange thing here is an ovum or egg, as we call it in slang, right? And so it shows what that looks like once it starts to fertilize. But this is not fertilized yet. This sperm has to be able to get all the way through and fuse with the membrane. So we'll be looking at that. Fertilization. So you have about, 300 million sperm that will be released into the vagina of the female, and it has to travel through the acidic vagina, which kills the majority of sperm. Then it has to get into the cervix, has to go through some cervical mucus, and then there's also some strong uterine contractions that will interfere with the ability of the sperm to reach the fallopian tube. So you only have about 10% of all the sperm that will even get to the fallopian tube. Of course, then it still has to travel through the fallopian tube and get to the ovum, and then burrow through and actually fertilize. Okay, once those survivors get to the fallopian tube, they undergo a process called sperm capacitation. So you've heard you say like, oh, I'm incapacitated. So you can think about that capacitation. Okay, so a couple of things happen. Actually, a lot of events happened, and your book does go into some detail if you're curious. But basically, the sperm get kind of activated, so they're going to start swimming quickly towards the ovum. It's like, oh, the finish line, the end is in sight. And it's not really in sight because they don't have eyes, but they have chemical cues, and they also have a temperature cue. So chemotaxis and thermotaxis. The acrosome also becomes fragile, and it's getting ready to for the next stage, we'll just say. Okay, so these are simplified steps of fertilization. It's actually amazing how much we know about it, and you could study fertilization for weeks. There's a lot there. So your book has a little more detail than we're going to go into, but you could even go through in much more detail than your book even presents. Okay, so sperm penetration of ovum. So it reaches the ovum, and it has to break through the outer layer called the corona radiata, which is made up of granulosa cells. The acrosome reaction then happens. So remember we said the acrosome becomes fragile, and that's good because if it breaks through the corona radiata, then the acrosome will have this special reaction that releases the enzymes that are stored in the acrosome, and it releases those enzymes to help it break through the zona pellucida. And if it gets all the way through the zona pellucida, then the membrane of the sperm, so that blue part in the picture, fuses with the plasma membrane of the ovum, and that releases the genetic material of the sperm into the ovum. So again, when you started out, you had this acrosome with this outer layer holding in all the enzymes. Then it gets a little weak and it's getting ready for the acrosome reaction. When binding of the sperm to the ovum happens, so binding to the zona pellucida, that second layer, that's going to stimulate this fusion reaction where you basically get these holes that allow the it's not really holes, but it's exocytosis reaction that allows the enzymes to be released, and that helps you to break through the rest of the zona pellucida and then fuse to the membrane of the ovum once you reach it. So then once you reach it, this membrane here fuses to the ovum and then releases the contents into the ovum. So nucleus of the sperm cell moves into the cytoplasm of the ovum, and then this will cause changes to occur to the ovum itself. And most importantly, well, all of these are super important, but the first thing that needs to happen is that you need to prevent any other sperm from being able to get to fertilize this egg, right? We can only have one set of chromosomes added to the ovum to make a human, okay? So we call this a block of polyspermy, so mini sperm, right? We need to prevent more than one sperm. And the video that I've included in the next slide or two will show this. Excuse me. I did warn you, I'm trying to wake up. Okay, so you're going to, the ovum's going to release peptides and other components that will harm any other sperm that have been trying to get into the ovum. It's also going to change its zona pellucida. You can actually see it like lift. You see this huge wave, this really dramatic action occur. So the block to polyspermy is a really cool event, okay, and super important. Then once we've taken care of that, the ovum knows, okay, it's actually happening. I've been fertilized. It's going to quickly finish its own meiosis too. So this is weird because the ovum actually had not finished meiosis 2 to become a complete gamete like sperm did, okay? So like I said, female reproduction is more complicated. Okay, so the ovum quickly finished meiosis 2, and we'll talk about that in the next lecture more. Then it's ready. It gets rid of that sister chromatid material and only has 23 chromosomes with individual chromatids, and then that will combine with the sperm's individual chromatids, right? And that combines, and now you have a complete set. You have one set from mom and one set from dad, which makes 46 chromosomes. But yeah, they don't have sister chromatids at that point, but they're going to then undergo replication, and then cell division is going to happen very rapidly. So as soon as the ovum and the sperm chromosomes combine, we call that a diploid zygote. Yes. And then the zygote undergoes division, and eventually it will be called, well, there's lots of different terms, but for us, we would think like it will develop into a fetus. Okay. Also, you know that the zygote right now is in the fallopian tube. It needs to travel into the uterus and implant into the uterus. Okay. So there's a lot of times fertilization occurs, but pregnancy does not occur, because if you don't implant, then development stops and they'll just be washed away with the next menstruation or period. Okay. So here is what we're talking about. You have the slightly weakened acrosome that contains enzymes. It starts to burrow its way through the outer layer, the corona radiata, the granulosa cells, and then that activates the acrosome reaction and releases those enzymes so that it can burrow all the way through the zona pellucida. Then it's going to fuse the membrane of the sperm, fuses with the membrane of the ovum, and that releases the sperm's nucleus, which I know it shows as just being the same, but it would be a component. But yeah, the majority of this is just genetic material, so that's why it looks the same here. And then that is going to sit and wait just momentarily. It happens pretty quickly, the reaction, to prevent any other sperm from getting in, and then the ovum has to complete its it's been suspended in meiosis. The second division has to finish, and then it will combine with the sperm and be a zygote. Here's a video, please watch it. It's a really good one and it's not very long. Okay, so successful zygote and implantation and pregnancy. Okay, so now you have a developing fetus. 2 of those 46 chromosomes are going to determine the sex of the developing fetus. If it has an XY chromosome, those 2 chromosomes, if it's X and Y, then it's male. If it's X and X, it's female. So this is the genetic sex. And the reason why is that this Y chromosome has a sex determining region. So it has genes that will stimulate the gonads to become testes. If you don't have the SRY, sex determining region, if you don't have the SRY, then the gonads will automatically develop into female reproductive gonads. Okay. What else? Yes. So you become, the Y chromosome has the SRY that signals the gonads to become testes, and of course the testes then make testosterone and sperm, right? So that you have a functional male. Okay. Females, of course, lack this gene. They have an X instead of a Y. The X does not have the SRY. So the gonads will develop into ovaries because they don't have the genetic material to tell it to do something else. Okay? So one more time. Sperm and ovum combine, and either you get XY, or if the sperm was carrying an X chromosome, you get XX. So all female ova are always X, right? We are XX. The only possibility that we have to give to our offspring is an X. But men are XY, so some of their sperm will be X and some of their sperm will be Y. Okay? So if you happen to be XY, then during development as a fetus, you will have the SRY genes that will signal the gonads to become testes. And then you will have seminiferous tubules, you'll have interstitial cells, and you'll develop, that develops early on in the embryo. Okay? So early in development, all the gonads are the same. And then at, what was it, week 7? Starting at week 7, then you start to develop the gonads more specifically into either testes or ovaries. And again, in order to make testes, you have to have the SRY region, sex determining region Y chromosome. Okay? So, more about male reproductive system. The hypothalamus releases GnRH, which you know stands for gonadotropin releasing hormone, and that travels and signals the anterior pituitary to release FSH and LH. And you recall that LH stands for luteinizing hormone and FSH stands for follicle stimulating hormone. And they were named originally discovered in females, so the names reflect more on the female reproductive, but men have it too. Okay, so you need to know the different role that LH and FSH play for men versus women. Okay, so memorize this, and then when we do our female reproduction, you'll see another one for female, and you'll need to memorize that too. Okay, so anterior pituitary releases LH. LH then signals the testes, and specifically the Leydig cells or interstitial cells of the testes, to secrete testosterone. That testosterone will then stimulate sperm production. But once you have enough testosterone, it will act as a negative feedback to tell both the hypothalamus and the anterior pituitary that you have enough. So this prevents too much testosterone. Okay. At the same time, the anterior pituitary, I keep trying to put those words together. Anterior pituitary releases FSH, which signals the seminiferous tubules of the testes to secrete inhibin, which is such a nice name because it does exactly what it's called. It inhibits the anterior pituitary, right? So every time you see this negative sign in a circle, that means inhibit or turn off or slow down, okay? Act against. So both testosterone and inhibin have negative feedback on the anterior pituitary, but testosterone also has a negative feedback on the hypothalamus itself. So if you want to be able to release FSH and LH, you need gonadotropin releasing hormone. Okay. What else did I want to say? And I did say, but it's not drawn here, that testosterone will then talk to the seminiferous tubules to turn on sperm production. Okay. So testosterone produced by the interstitial cells of the testes, right? We just saw that right here, right? Leydig cells, okay? And it is an anabolic steroid, right? It's a sex hormone, but it's also, it's a steroid hormone, and we call it anabolic because it stimulates growth of all sorts of tissues, right? It turns the boy into a man, right? So you get an increased muscle mass, you get a deepening of the voice, you get facial hair, pubic hair, right? You get sweat glands become more active. You start releasing more oils, right? So all sorts of development. Okay, so pretty much all of the growth and development is in boys is testosterone. Okay, it also signals sperm production in the seminiferous tubules. Okay, so question for you. A 38-year-old male is upset about his low sperm count and visits a practitioner who commonly advertises his miracle cures of sterility, the inability to have a child, right? The practitioner treats conditions of low sperm count with mega doses of testosterone. The patients have a surge in libido, and that means their sex drive increases. However, their sperm count actually gets lower. So we want you to explain why. So this practitioner that's giving these mega doses of testosterone is not legitimate, right? Because it didn't work. It doesn't work to raise sperm count, and it's not going to treat sterility. So what would a legitimate practitioner do for a patient? Pause the video, work through all the parts of this, and then come back for the answer. Okay, I trust that you paused it and you worked through this for a while. Okay, so recall our negative feedback loop. So what would mega doses of testosterone do in addition to, at first it does stimulate sperm production, but it also turns off the anterior pituitary and the hypothalamus. So having a huge increase in testosterone turns the whole thing off, and now you won't get any extra sperm, right? You're gonna have less. It's gonna drop. It's gonna make the problem worse, right? So what should have happened? A real fertility doctor would first take blood work and a semen sample, and would look for levels. So in the blood work, we're looking for the hormones, LH, FSH, GnRH, right? We can give gonadotropin releasing hormone if that's what the problem is. We can also give FSH or LH, right? So whichever, if there is an imbalance, if there's a low level of one of these hormones, we can treat it with supplements, shots, right? But we would also look for other factors, and we would also do a little counseling, right? So we would ask about lifestyle. There are simple things that can be done, like if you go to a hot tub before you have sex, the high temperature of the hot tub is going to kill the sperm. Now, please don't rely on that as birth control. It's not 100%, right? But it is actually pretty effective. Tight underwear can also interfere with sperm. Certain drugs will have an effect as well. So a real doctor would examine the entire picture, find out what's going on, and then treat the cause, right? Also, if libido was an issue, then you would recommend things like stress management, which is usually exercise, but can also include meditation and other practices, right? So stress management, and then also relationship Are you bonding with your partner? Do you feel safe? Do you feel loved? Because those things are going to also play a huge role in your libido, right? If you don't feel loved and you don't trust the other person, you're probably not going to want to reproduce with them. Okay, so that's it. That's the end of this lecture, and I'll put up your the next one for female. Get ready, it's more complicated. Okay, and then that's it. Yay.